Pathology, the principles of neoplasia. All right, so neoplasia, once again, is just another fancy word for cancer. And what we need to understand is how that occurs. So first off, it is an irreversible, uncontrolled, monoclonal proliferation of cells. So I want all those things to be said and heard individually because they're all very important. Once again, this is irreversible. So once we progress to neoplasia, there's no going back, okay? There's no changing cancer. Cancer is what it is at the end of the day, sadly. It's uncontrolled. We have no ability to control it. We could just try to kill off all reproducing cells, right? Replicative cells, but there's no way that we could genuinely control or slow down that production, right? And lastly, it's monoclonal, which is the most key factor to this. It all stems from the proliferation of one cell, okay? The proliferation of one cell, then it continues to proliferate into multiple, okay? But it comes from that one cell lineage, which is so important and very interesting when it comes to cancer, okay? Now, what you have to understand is the five main steps in which everything occurs. So the first step is just normal cells. Okay, so we have normal cells located with the epithelial cell layer. We have the basement membrane, which separates it from the blood and lymphatic systems. Okay, now then we have loss of uniformity. Right, there's multiple. They call it pleomorphism. Okay, so we have loss of cell size and shape uniformity. Okay, so you will see different sizes, different shapes of cells, and this is known as dysplasia. And we already discussed in the cellular adaptations that this is a reversible type. Okay, this dysplasia is reversible because it really hasn't broken through and hasn't been invaded into our basement membrane. Next, we have carcinoma in situ, okay, which is pre-invasive, but it's irreversible. Even though it hasn't invaded yet, it's still irreversible because of the amount of replication that we've acquired, okay? Next step, fourth step, is our invasive carcinoma. Our cells have finally broken, our cancerous cells have finally invaded and broken through the basement membrane. And once again, a key concept to that is the usage of tumor cells using collagenases, also known as metalloproteases, okay? They love that. They're gonna use them interchangeably. You have to understand that they use collagenases and metalloproteinases, okay? And what do? They also attack the E-cadherin, okay? By attacking E-cadherin kind of links cells together. Once they break E-cadherin apart, they're able to grow between cells and get down to that basement membrane. Once at the basement membrane, they're able to use collagenases and metalloproteinases to break through that collagen type 4 and get through our system into the blood and lymphatic vessels to go to our final step, number 5, for metastasis. All right? So knowing that process is very important. I'm going to start over real quick. You have normal cells, then you have dysplasia with the loss of uniformity due to pleomorphism, which is reversible. Then you have carcinoma in situ. Essentially everything is cancer, but you haven't invaded, you haven't broken through the basement membrane. Then you break through the basement membrane and it becomes invasive carcinoma due to the fact that you have collagenases and hydrolases, and you also have inactivation of E-cadherin. And then lastly, you have metastasis when you get into the lymphatics and the blood. Okay. So tumor nomenclature is really, really important for understanding how we define what's going on. Okay. Now, the book kind of goes through a lot of different things. I'm just going to go with the quickest and easiest things of understanding what happens. There's two types, carcinoma and sarcoma, okay? For me, the best way to remember it is clash, okay? They like to use clash all over the place. So what that is that you have CL goes with together and then SH goes together, CLASH. So CL's together, SH is together. Now, what does that mean? So carcinomas travel mainly via the lymphatics. So lymphatic spread is known as carcinomas, okay? When you have hematogenous spread or spread through the blood, you have sarcomas. So C and L, carcinoma in the lymphatic spread, and then SH is for sarcomas in the hematogenous spread, okay? Now, that's not a hard, fast rule. It's just a way to remember kind of the majority of what we're gonna see. Now, there are some exceptions, and here we are. The exceptions are follicular, choriocarcinoma, renal cell, and hepatocellular carcinomas, okay? So those 4 carcinomas, they do not travel via lymphatics, they travel via the blood, okay? And they have a very great mnemonic, and it says, it goes, 4 carcinomas route hematogenously. So 4 starts with F, that's for our follicular carcinomas. Carcinomas is choriocarcinoma, starts with a C and C. Route, R, is for renal cell carcinoma, and hematogenously is for hepatocellular carcinoma. This is good in 2 ways because 1, it's a mnemonic, and also it tells you what the mnemonic's for. The name says 4 carcinomas route hematogenously. It's telling you which 4 carcinomas are the exception to the lymphatic rule and actually travel via blood spread, okay? That's the nomenclature that I care about, and that's all that you really need to know, okay? Moving forward, we're gonna talk about tumor grade versus stage. When you have a high grade type of tumor, it's poorly differentiated, okay? So it looks completely different from the native tissue, and that's a poor prognosis, okay? That's what they're saying. High grade is poor prognosis, bad, okay? Looks different from native tissue. If they say low grade, that means it's well differentiated, okay? So that means it resembles somewhat normal tissue because it's differentiated. So that is a better prognosis. So low grade, better prognosis, high grade, poor prognosis. Moving on to stage, okay? So stage is mainly dependent on whether we have distance of metastases, okay? So how far we have spread the cancer. So if you have a higher stage, that means that we've just spread further and it's worse. If you have a lower stage, you haven't really spread that much, okay? It could potentially be benign, okay? And not have spread anywhere. Got it? And so a lower stage is better, a higher stage is worse, okay? Now, the hallmarks of cancer are rather important. And so I'm just going to go through this whole entire page and kind of break it down for us, okay? So the first thing we need to understand is how cancer really acts, okay? It favors chaos, okay? So think about just this cell that says, hey, I'm going to break all the rules to what we understand about what a normal cell should be. I want to replicate as fast as I can. I don't care about checking for any mutations because more mutations means more issues, means more problems. I don't care. I just want to replicate as fast as I can and produce, okay? And so any type of DNA repair, any type of known survival, right? If we're trying to get as much, oxygen in that area, we're producing any type of reactive oxygenases species, it doesn't matter, okay? We're going to be using glycolysis in an anaerobic state. To us, whatever matters is just reproduction. Cancer is just trying to live, weirdly enough, okay? So that's how I think about it. Cancer is just trying to live. It's going to be doing anything in its power. It doesn't care about DNA mutations or anaerobic production via glycolysis. It doesn't matter. It just wants to reproduce, okay? So the first thing we have to understand is our mutations in tumor suppressor genes versus proto-oncogenes, okay? And there's going to be a larger chapter on that later on, but I just wanted to point it out. So there's proto-oncogenes and there's mutations to our tumor suppressor genes, okay? And so those are going to be really important for tumor genesis later on, and we're going to talk about it later on, okay? Now, what do I care about? Loss of E-cadherin, we already discussed. So the loss of E-cadherin allows for cells to no longer stick together because E-cadherin brings our epithelial cells together. If we have the loss of E-cadherin, our tumor cells are able to go in between the cells and reach the basement membrane to use collagenases as well as metalloproteinases to get through that basement membrane, collagen type 4, okay? Important. Next is that they have the ability to evade apoptosis. How do they do that? They have mutations in their genes that regulate apoptosis. So we already discussed BCL2 as a very key factor to this. BCL2 doesn't allow for cytochrome C to be released from the mitochondria in order for apoptosis to occur. Well, a cell that senses that they are cancerous, they're going to probably want to do that. They want to, activate apoptosis to go and kill itself. Well, at the end of the day, tumor cells have mutations in order to get around that, and they have an constantly active BCL2, okay? And that's where we'll see follicular B-cell lymphoma play a role in that, okay? Next is limitless replicative potential. So what they have is the ability to reactivate telomerase. So as we age, and as all cells age, their chromosomes have these little things at the ends known as telomeres. And as we age, those telomeres shorten and shorten and shorten. But what we've noticed is that stem cells, okay, have this thing known as telomerases, which stop the shortening of those telomeres, okay? However, we've also noticed that tumors have those same telomerases, okay? So it leads to the potential for longer life, but also leads to or yields to tumor genesis, okay? So very important because it prevents chromosome shortening and it blocks that cell aging, which can lead to the tumor genesis, okay? Next, it likes those increase in VEGF, right? Pro-angiogenic factors, and it decreases inhibitory factors. The reason behind it is because it wants angiogenesis. It wants all the blood it can get. It wants reproductive factors, and it also wants to spread via the blood vessels. So it's going to attract that VEGF in order to produce more vasculature, okay? Next, it can use immune evasion in cancer. So cancer has immune evasion properties. What does it do? Well, it decreases its MHC class 1 expression. So normally, all of our cells in our body have MHC class 1. And what it does is that it will present to the environment, or antigen-presenting cells, hey, I'm sick, okay? So, hey, this cell is producing its MHC class 1. It's telling the environment, hey, there's something wrong with me. So then the CD8 cells, the cytotoxic cells, come and they bind, they sense the MHC1 class, and it's going to kill that cell that is sick. Well, cancer decreases the MHC class 1 expression, leading to an inability for cytotoxic T cells to kill them. Okay. Next, it secretes its own TGF beta and IL-10, which we know slows down the immune system. It's anti-inflammatory, anti-immune response. Okay. So it recruits that regulatory T cells and it downregulates our immune response. Next, the hallmark of cancer is going to be tissue invasion. And you'll notice that I'm saying this over and over again because I believe that it's a really important topic. Once again, we have that loss of E-cadherin function. Once we lose that E-cadherin, our cells are now looser apart. They don't stick together very well. And so our tumor cells can get in between the cells, reach that basement membrane, produce collagenases as well as metalloproteinases, and break through. Okay? And that's a part of our metastases as well. So overall, in this small session, we've covered the tumor nomenclature, grade versus stage, as well as hallmark of cancer.